Hello, everyone, and welcome to our Bronx River Sound Shore Audubon speaker program with Violet Brill this evening, titled From Bronx River to Bear Country, a Student's Ornithological Odyssey. My name is Leslie Brill. I'm the chapter president, and the student in question happens to be my daughter. And she was responsible for getting me involved on the board and not the other way around. She was a youth representative on our board for many years. And I'm going to read her bio before I turn it over to her. So we will have a chance for a question and answers at the end, and always feel free also to type your questions and comments into the chat because we'll be reading them off at the end also. So, Violet Brill started birding with Bronx River Sound Shore Audubon when she was 10 years old with me and grew up co-leading foraging tours with her father, wildman Steve Brill, teaching about wild edible and medicinal plants in the New York region. After transferring from UMass Amherst Honors College, she is now a senior at Cornell University's College of Agriculture and Life Sciences, studying environment and sustainability, with a concentration in environmental biology and applied ecology. Violet conducts research with Jen Walsh in the Fuller Evolutionary Biology Lab at the Cornell Lab of Ornithology, where she combines genomics and field studies to understand the ecology, behavior, and conservation of songbirds. She is passionate about ornithology, conservation biology, and environmental education. So we are excited and honored to have Violet back tonight. One last note, she's sort of back by popular demand. About 2.5 years ago, she did a speaker program for us on early spring foraging. And if you'd like to watch the recording of that, you'll find it among the archived programs on our website. So Violet, take it away, Violet.
Sure. All right, I'm going to share my screen. Let me just up here. Okay. See. Um, can everyone see my screen?
Yes.
Yeah.
Okay. Perfect. So hi, my name is Violet Brill. Thank you all for welcoming me here today. I am so excited to talk to you all about some of the research that I've been doing, as my mom said, from Bronx River to Bear Country, a student's ornithological odyssey. So to begin, I have been foraging my entire life. This is my passion. Me and my dad lead foraging tours in the New York region, and we teach people how to identify wild and edible and medicinal plants and teach them how to use these
Must
plants
have puked all over your nice tablecloth.
for food and home
Huh?
remedies.
All right.
And this is something I'm very passionate about. So I'm passionate
Who is
about
you watching Violence?
environmental
Uh-huh.
education
Always haven't seen
and
that.
yeah. So this is something that I have been doing my whole life, ever since I was very young. So this is really what got me passionate about nature and the environment in general. And it was when I was 10 years old that I began birding
You're
with the Bronx
recording. You
River
can
Sound
modulate.
Audubon. And I really wouldn't
You
be here
gotta
talking
take
to you
care of that.
all
Yeah.
today if it weren't for the Bronx River Sound Shore and all these wonderful people in this organization. So this is really good what got me started and passionate. And it was actually with Sandy that I first had my experience with bird banding, and I have very fond memories of banding bluebirds with Sandy when I was like 10 and 11 years old. And I also ended up getting a parakeet who I named Wisteria. So yeah, this birds are something that I've been passionate about for many years. But today, I'm going to be talking to you about some recent research I've been doing and that I've been so fortunate to be involved with that has taken me to British Columbia, Canada, and also to Montana. So yeah, just to give you a little bit of background, which my mom already kind of did, but I started off at the University of Massachusetts Amherst and I transferred to Cornell University. When I transferred to Cornell, I was able to take Dave Bonter's field methods class, which really gave us just a broad introduction on these different methods for studying birds in the field, from point counts to measure bird abundance to sound recording and listening to their songs. The first, um, there we go. Yeah. Okay. The first, um, the first time I was studying, collecting data in the field, was over a spring break trip to Organ Pipe Cactus National Monument, where we were collecting data on verdin and their nesting success and their reproductive success, contributing to a long-term field study. And then, of course, I was able to, the following summer, travel to Mandarte Island, which is where my senior thesis research is focused on. And finally, this past summer, to the University of Montana's Bird Ecology Lab, where I was a field technician, helping a master's student on one of her projects. So just to give you a kind of overview of what I'm going to be talking about tonight, first I'm going to be talking about a little like side project that I've been working on for a while on this novel nest construction behavior in Sooty Fox Sparrows. Um, most of this is going to be about fox sparrows. And then I will take you through some of my senior thesis research, which is focused on variation in Sooty Fox Sparrow migratory behavior. And finally, I'll talk to you about some of this ecotoxicology work that I did in Montana over the past summer. So, Mandarte Island. I was very fortunate my sophomore year, I'm now a senior, to connect with Jen Walsh, who's a research associate in the Fuller Evolutionary Biology Lab at the Cornell Lab. And she invited me to complete a field season on Mandarte Island, this island where there's been a long-term study on song sparrows for many, many years. And we were also guided by Peter Arcese, who ran this project through the University of British Columbia for many years before my advisor, Jen, recently took over. So, Mandarte Island is this very small island located just off the coast of Vancouver in British Columbia, Canada. It's really only about 700 meters from one end to the other. And so, as the Google Maps name suggests, Bear Island 9, there's not much on it besides these sparrows and their habitat and all the stuff that we really, really care about. Um, so yeah, there's no running water or electricity on this island. Me and Miranda were there for 10 weeks. The island's also named Zoxtel, which is the indigenous name. It is land of the Tsuut'ina First Nation as well. So Mandarte Island is home to the song sparrows and fox sparrows, but of course it wasn't always that way. The Mandarte Island project is centered around the song sparrows. It's a small population on a small island, so it kind of opens doors to ask interesting questions about the evolution, about how these species are locally adapting to their conditions, assessing the genetics of this population, the demography of the population. And so it wasn't until 1975 when the Sooty Fox Sparrows colonized the island. And when they colonized the island, it was people noticed them and they began banding them. So for every song sparrow on the island, we know every individual song sparrow that has ever bred on the island and every chick that has ever hatched on the island. We don't have as much data on the fox sparrows. And so the fox sparrows are really this kind of unknown, like untapped source of research, I guess. There's been a little bit of work done on them, but we really don't know much about them, at all. So we were pretty interested in doing a project on the fox sparrows as well. So with the dynamics of these two species on the island, just to give you like a brief background, The song sparrows, so the song sparrows have been studied since the 1960s. Like this is for over 50 years. This is a really long-term project, and their population has been known to go through kind of these boom and bust cycles. But when fox sparrows colonized the island, the song sparrow population began to decrease. The fox sparrows are the larger of the two species, and they're the dominant of them as well. And so as fox sparrow population increased, the song sparrow numbers of breeding females and juvenile overwinter survival decreased. So me and Miranda were on this island, and Miranda's also another undergraduate. And so we were trained on the island. That's Catherine, training me with a sparrow. We were responsible for monitoring territories, nest searching, banding and bleeding chicks, capturing and banding unbanded adults, and also mapping the territories of these individual birds. Here's just some pictures of the maps that we used at the start of the field season. The map of the island is kind of split up into all the different song sparrow territories, and then they each have a unique color band combination. So these little color bands that they put on the bird's legs so we can identify each individual. But what really interested us about the fox sparrow population at first was their nests. So what's unique about this is that they line their nests with these white down feathers from the glaucous-winged gull colony.
Violet.
Yes. Hello? Okay. From the glaucous-winged gull colony, which also inhabits the island. And this is a really unique behavior. It has not been documented in any other fox sparrow population. They don't typically line their nests with feathers at all. And this has consistently been observed ever since their colonization on the island. So this made us wonder, what function might white glaucous-winged gull down serve in Sooty Fox Sparrow nests, and has this behavior been documented elsewhere? So we know that feathers can play important functions in bird nests. We know that they can provide insulation to nests. They can play a role in signaling phenotypic quality and like good genes. They can maintain a microclimate within the nest, and they can also defend against harmful microorganisms. So I went searching for any other documentation of this behavior. I went on VertNet, and VertNet is basically an online database that is just a collection of all museum specimens. So it includes nests, it includes actual birds collected as specimens. It's just a really great resource. I also went on the Macaulay Library and peer-reviewed literature as well. And so we found a lot of nests, and really no others except the one in the bottom right corner that also had white feathers lining the nests. And no other nests contain, we have one mention in the literature from a really old paper of a fox sparrow nest that contained feathers in it, but they don't typically line their nests with feathers. So the first thing we noticed is just a ton of within species, so interspecific variation in fox sparrow nest construction, which you can kind of see from these pictures that they are all quite different from one another. Some are lined with more dry grass, some include bark, some include moss, dry leaves, and then the white feathers. So we found that there was also a differences geographically in the distribution of what lining materials were used in what areas. And so we were sticking to just Western North America in our research. And you can see at each location there's a lot of variation in what material is used to line their nests. But in British Columbia, which is just our Mandarte population, all the nests contained white feathers. This is also just a bar plot showing the percentage of nests containing each lining material that we found. Grass is the most commonly used material, but we found some records of other materials being used as well in nest lining. So I went searching, and in my research, I found a few papers, and they were talking about barn swallow nests and tree swallow nests, and the possibility that these feathers that they line their nests with might be useful in defending against microorganisms and harmful pathogenic bacteria. They were looking at the different colors of the feathers as well and seeing if there were any differences. And so I found some pretty interesting results from my literature review that helped me formulate my hypotheses about why the fox sparrows might exhibit this behavior. So in terms of results, the sooty fox sparrows on Mandarte really exclusively line their nests with the white gull down. And what happens is with any feather, a feather once it's off of a bird is usually colonized by some type of feather degrading bacteria. And that's just involved in the decomposition process of the feather itself. Um, these feather degrading bacteria degrade the keratin that feathers are made out of. And some of them, such as this Bacillus lanciniformis, actually produce antimicrobial substances in order to outcompete the neighboring bacteria also trying to grow on those feathers. Um, so this antimicrobial so this is important. And this feather degrading bacteria actually grows better on unmelanized, and melanin is is a pigment, is what gives feathers like a dark pigment. And so we found that this feather degrading bacteria grows more on unmelanized white feathers than it does on darker pigmented feathers. So I'm thinking that by lining their nests with these white feathers, the fox sparrows are actually might be adapting to the local conditions of Mandarte, and of the bacterial environment of the island, controlling kind of the microbiological environment of their nest. There's some harmful and pathogenic bacteria that can lead to trans shell. It goes through the shell and it infects the embryo before it hatches, and that it can reduce hatching success. There's also some bacteria that can colonize nestlings and reduce fledging success. And so, if these birds are lining their nests with these white feathers that have antimicrobial substances, they might be defending against this harmful bacteria and thereby increasing their reproductive success. So, so I presented this research at at AOS, the American Ornithological Society conference in St. Louis in August, which was lots of fun. And actually, it was just today we wrote a natural history note based on this, and we actually just submitted it to the American Naturalist Journal for publication. So who knows what's gonna happen? We don't, but we just submitted this for review. So um, we'll see what happens there. But that's that project. That's one of the things I've been working on. And the next thing I'll talk to you about is my senior thesis research, which is again focused on the fox sparrows on Mandarte Island. And it's titled Genomic Architecture Underlying Migratory and Resident Life Histories in Sooty Fox Sparrow Subspecies. So to just zoom out for a second, fox sparrow, there are four groups of fox sparrows. We have the Iliaca, which are the red fox sparrows, which we have here. And there are three other groups. The ones that I'm studying are the sooty fox sparrows, which is the Unalaskeensis group. And I'm not sure if you can see my cursor, but their range extends from in this dark gray from just around Vancouver, and it hugs the Pacific Northwest up through Alaska. And what's important is that sooty fox sparrows, the Mandarte Island sooty fox sparrows, are one of the only resident populations of fox sparrows. Other populations are migratory, whether they're short distance or long distance migrants. So, oh, here we go. So there's a lot of variation in fox sparrow migratory behavior. In the east, we kind of have this more typical breeding up in Canada and wintering down south. But in the Western populations, we have some fox sparrows that make these transoceanic flights from up in Alaska down to California. We also have some of the fox sparrows in the West exhibiting this migration, engaging in this leapfrog migration behavior. And fox sparrows, the sooty fox sparrows, are typically known to engage in an altitudinal migration. But as you can see on the map, some of the ones around Vancouver and Vancouver Island are a mixture of migratory and then some year-round resident birds as well. So I was pretty interested in just this variation in migratory behavior. And if you think about it, it makes sense that the fox sparrows on Mandarte Island would be a resident population. They're able to fare quite well there. They subsist on seeds during the winter time. And this is one of the reasons that the fox sparrows on Mandarte are able to outcompete the song sparrows so successfully. It's because they're outcompeting them for food and for seed during the winter time. And migration is a strategy that allows species to follow fluctuations in vital resources. But if the fox sparrows on Mandarte have their food source year round and they can stay and conditions allow them to stay on Mandarte Island year round, why would they waste the time and energy in migrating? And migrating doesn't just involve the journey itself, but it involves the preparing to migrate. It involves refueling along the way, and it's just a generally very risky process. So why would you waste the time and energy in migrating when instead you could invest your energy in perhaps extending that window and that time period during which you're able to reproduce? So I think it makes sense that they would be residents. This is a study done by Hannah Visti, and she was comparing fox sparrows that were migratory in Alaska, and she was looking at the demography to residents on Mandarte Island. And so what she found was that, as you would expect with a resident population as compared to a migratory one, the residents exhibit a longer breeding season. They initiate breeding earlier, they produce more broods annually, and they lay smaller clutches. So the trade-off is very clear here. They're instead of investing energy in migrating, they're extending their breeding season. So just to pivot a little bit, just to give you a background on genomics, because this is pretty central to this project. So I'll take you back to biology class for a moment. But genomics is really the study of an organism's whole complete set of DNA, and its DNA is known as its genome. DNA is this, as you see in the top right, this double-stranded molecule that is composed of these different four chemical bases: adenine, cytosine, guanine, and thymine, which make up genes and a bunch of other stuff as well. But through genomics, you can look at how genes function and interact and evolve over time. Through DNA sequencing, you can determine the order of these four nucleotide bases. And from that, we can understand how genetic information shapes certain traits and responses to the environment. So from genetics, we can ask, oh, what is the sex of an individual? We can ask what genes control plumage traits, what are the family relationships between individual birds. So I got involved in the Fuller Evolutionary Biology Lab, which is really focused around genomics, which is what led me to the project that I have. I kind of wanted to take a different genetics twist on Hannah Visti's paper. But so I was interested in this variation in migratory strategies, and my research question is, what is the genetic architecture underlying observed differences in migratory behavior in sooty fox sparrows? And I'm also wondering, how might this inform our understanding of their ability to adapt under environmental change? So I'm thinking about how might this variation in these behaviors, what might that tell us about how resilient the species can be when faced with environmental change and climate change? Especially thinking about Mandarte Island, which is in the Southern Gulf Islands in Canada. It's kind of in a vulnerable location when you think about this environmental change. So I think that this information from like this can help us better make predictions about how species distributions might change in the future, and also how a species just might survive in the future in general. We can predict what their population trends might look like and their general survival. So again, I came back to VertNet because I was interested in comparing a migratory and resident population. So I would need a migratory population to compare the Mandarte individuals to. So again, I went on VertNet, which is just this online database of all museum specimens. And I was looking for tissue samples from migratory individuals in Alaska. They had to be they had to be recent samples because DNA degrades over time, and it turned out that there were a number of samples, and those were at the University of Alaska Museum of the North. And my advisor, Jen Walsh, has connections with Kevin, who works there. And so they were very generous, which I'm very grateful for. They actually provided me with tissue samples to use for my project. And once I had these tissue samples, I was able to apply for funding through CALS and through the lab as well. And I'm so grateful for this funding because it really allowed me to proceed with this project. Just to give you a better understanding of where my samples are coming from, we have the Mandarte Island individuals, and then we have the Kodiak, Alaska individuals. And if you recall the range map of the Sooty Fox Sparrows from earlier, you remember that these are kind of the extremes of the Sooty Fox Sparrow breeding range, which works out quite well. So I'm using a whole genome sequencing approach. And there's me bleeding a fox sparrow. Instead of just looking at a specific part of the genome, because we don't know what genes might be involved in migration and where they are in the genome. So we're sequencing the entire genome, which contains billions of these nucleotide bases. And I have my 10 blood samples that I collected on Mandarte and the 10 tissue samples from Alaska for a total of 20 birds. So I began a literature review. Do we know anything about the genetic basis for migration? Is there a genetic basis for migration? Or do we know what genes are involved? And it turns out that, we know a little bit. There's some conflicting results, but yes, there is to an extent a genetic basis for migration, and there's some genes that have been proposed to play a role. And genes that are proposed to play a role in a trait are known as candidate genes. So just to kind of go over how I'm thinking about migration and also just some notable findings from my literature review. I think about migration as this integration of behavior, physiology, morphology, and life history. And it's useful to kind of break it down into its three components: timing, direction, and distance. And so genetic factors can play a role in these three components in some or not all, depending on the species. So, as we all know, migratory behavior is highly variable within and among species, and much of this variation is due to genetic differences. We also know that some past studies have identified genes that might be candidates in playing a role in either the trait directly or the variation that we observe within or between species. And just an interesting hypothesis. For a hot second, we thought, oh, maybe there's just one gene that controls migration, and we know that's not the case. It was hypothesized that it's hypothesized there exists maybe a migratory gene package, a set of genes that are inherited together that impact the observed phenotype. And I mean, my results don't really support this, but I just thought that was an interesting hypothesis. But so I got into the lab and I spent many, many hours in the lab, and I won't get into the details too much, but basically what you have to do for a project like this is first you start with extracting DNA from your blood or tissue samples, and you make genomic libraries which end up in tubes. And this is just basically an each individual, it's DNA cut up into a bunch of little pieces, like barcodes added onto them so that you can identify what individual each piece of DNA came from that's in a specific size. And then with sequencing, you end up with like a machine like this. Each of these columns are like a lane, and so the DNA, each like base, each nucleotide gets like a dye color on it. And when they sequence it, they shine like little fluorescent lights on the DNA. And from those fluorescent lights, they can tell what is an A, what is a T, what is a C, and what is a G. And you can tell the order of your bases, from which you can tell where there are genes and how the genome is structured and like the architecture of the genome. But basically, whole genome sequencing, you don't need to know what this means, but it resulted in 20 times coverage. And I'll talk about what a SNP is. Over 9 million SNPs. Basically, this is just really high quality data, which I was very glad to get pretty high quality data to work with for a project like this. So we have our data from our Mandarte sparrows and from our Alaska sparrows, but you need to compare that data to something. So you have a reference genome. And this is, you can look at this like horizontally. It shows like chromosomes, and it just shows like this green, like the thin green bar going across is like one gene. We're using a swamp sparrow reference genome just because it was annotated really well with where there are specific genes, and it's a really high quality gene, high quality even though they aren't like super closely related to one another. Yeah, to get into some of my analysis and results, we kind of have analysis in these four categories of just like population structure, like genome-wide statistics, and then we have my candidate gene analysis where I'm like looking at genes, and I'll explain these, selection scan as well. So, principal component analysis is really like the first step in a project like this, and it kind of just shows you how your data separates. In this case, like how my populations are separated from one another. So we know in theory, like we got 10 samples from Alaska, we got 10 samples from Mandarte, but we still like we need to see in the data like are we seeing what's expected, and it turns out that we are. So you see these two clear population clusters where Kodiak is separated on the left of the principal component one and Mandarte on the right. This next graph, you don't really need to see it. You don't need to know it. It basically it's the exact same data as before. It's just kind of funny because it's known as a scree plot, and so it again shows that the variation in the data is explained by one principal component. But I found out that this is why it's called a scree plot because of the mountain, which I just thought was kind of funny. Okay. So just to explain a couple of concepts before I show you some of my other results, FST is known as a fixation index. Like this is one of the main things that I'm using to see if to measure the genetic differentiation between these two populations. So how different are they from one another? An FST of 0 means that they are very, very alike. So you can imagine if you have two populations and there's a lot of movement between them and migration between them, there's going to be a lot of interbreeding, and those populations are going to be very similar to one another. Whereas an FST of 1, they're highly differentiated from one another, and those would be like very separate populations. And also a SNP is a single nucleotide polymorphism. So like if you think about like genome, as like a very, very long book with lots of pages. Um, there'll be like random mutations occurring over time. Like a random page in the book will like change over time. Um, and so on the genome, a change in like an A or a T, just these single base, like single nucleotide changes, these mutations is what a SNP is. So with that, this is a Manhattan plot, and it shows like all of my SNPs. So I have 9 million SNPs, and really the way to interpret this is just from the FST. So 1, 2, 3, like these are the numbers of my chromosomes. So this is like the whole genome from left to right, and it's showing where in the genome there are very differentiated areas. So you can see like with the really tall peak on the right at this spot around chromosome like 18, it looks like that spot is highly differentiated between those two populations. So my genome-wide average is 0. FST is 0.13, and that's actually a pretty high FST. And so it's really clear that these two subspecies within the Unaliscensis group are pretty differentiated from one another. Yeah, just to give you a recap on natural selection. So, natural selection is a mechanism of evolution, and it occurs when there's like a certain trait in a population that helps some individuals survive better than others. So there'll be different levels of fitness um, two between two like phenotypes or two morphs. So a very classic example is the pepper moth. After the Industrial Revolution began and there was like soot everywhere, the phenotype that was the dark color spread throughout the population because they were able to better camouflage in with that soot, and they were able to survive, and so that trait spread throughout the population. So in my data, there's a lot of like background noise. So one of the ways that I'm getting around this is a candidate gene analysis. A candidate gene analysis was just me really going through the literature, and I was just picking out genes from the literature that have been known to be involved in a trait that is important in migration, or it have been involved in variation in migratory strategies. So like there's a lot of work that's been done on like black caps in Europe because they have super long distance populations, long distance migratory populations. They have partially migratory populations, and they have like completely resident populations. And they associate a lot of those differences with differences in genes controlling their circadian rhythm. So a lot of those candidate genes are interesting to look at. But I'm going to talk to you about a couple that I found. And so what I'm trying to do with this is I have these metrics that signal differentiation between populations like FST, and I'm trying to associate these signals of differentiation with migratory traits, or like a trait that would be involved in a resident population like reproduction. So the first gene I'm just going to talk about is this metal 25 gene. And from this plot, the first thing I noticed is just how high the FST is. So it's about 0.96. So that means that at this location in the genome, there's really high differentiation between the two species. The way that we determined the significance of this was just that we have a smoothing line, and it peaked above 0.26, which is twice the genome-wide average FST. And then I was talking about natural selection. There's this metric called Tajima's D, and this is kind of indicative of natural selection in a population, like where selection might be occurring, and changing the kind of makeup of the genetic makeup of that area in the genome. So the more negative a value, that indicates selection is occurring there in the population, in that spot in the genome. So in the Mandarte population, we see that there is selection. It goes all the way down to -4 at the spot of this gene in the genome. And comparatively with the Alaska population, the line is pretty smooth. So just to recap with that, I have this gene, metal 25, and it's associated with nocturnal flight, and it's also associated with migratory timing in purple martins and in the great reed warbler. And so we're seeing this high level of differentiation at this spot in the genome between populations. And we're also seeing signatures of natural selection acting upon my birds from Mandarte. This other gene, Cry2, also involved in migratory timing. Its smoothing line peaks above our threshold. It has a moderate FST, about 0.75, but we can see from our Tajima's D that there is a really strong dip. So natural selection is acting upon that area of the genome where the gene is located, and comparatively, the Alaska population, it's really smooth, and it's really contrasting that. So just to recap that one, Cry2 is a gene that's associated with migratory timing. It's a member of the like clock gene family, so it kind of plays a role in the control of the circadian rhythm, which we know has which we know is associated with migratory phenotype. We're seeing moderate differentiation between our populations, and then we're seeing signatures of selection similarly on the Mandarte birds, as we did with the other gene. So that's kind of where things stand with this project. Before I move on, I'm just going to be finishing up my data analysis. I'm looking at, I have this candidate gene analysis, but what I can also do is I can look kind of from the outside in and look at the entire genome and see where is selection occurring on the genome, and then see if there are genes there that might play a role in migration. But I'm going to continue to write, and then in the springtime, I'm going to be submitting to target journals such as Ornithology, Evolution, and Molecular Biology. And so that is the second portion, and then we'll leave time for lots of questions at the end. But the last thing that I'm going to be talking to you about is my experience in Montana over the summer. So I was very fortunate to be able to work with the University of Montana's Bird Ecology Lab over the summer as a field technician, helping a master's student on her project, and I was nest searching. These are some of the nests that we found. I'll tell you what species they're from in a minute. So in Montana, this is kind of what our field sites looked like. They were often in gulches, which is just this kind of valley between the hills, and there's often a lot of wetland habitat there. It's also actively, like, these areas are actively being restored. But yeah, there are a lot of like streams. We used to have some beavers moving in. And our focal species, the yellow warbler, dusky flycatcher, warbling vireo, American robin, and Swainson's thrush. So I was working in the Mount Hagen injured area, which is the largest Superfund complex in the United States. And that's because of mining that's been going on there for a really long time. So I was living in a town called Anaconda, Montana, and Anaconda is named after the mining company who was there for over 100 years. And this is our smelter. So the smelter, there's mining for copper and gold in the area for a really long time, which is what the smelter was also partly used for. And so it's pretty tall. Those are buses on the bottom. It is actually taller than the Washington Monument. So they were mining in this area from the 1880s until 1984. So this smelter was really just huffing and puffing fumes with a lot of heavy metals in them for just about 100 years. And it had a really strong impact on this area's ecosystems, the structure of their ecosystem and just the ecosystem functioning in general and the natural processes taking place there. In other areas in Montana, you have really like dense like coniferous forests. Here, land is pretty, there's a lot of like barren land, and it's been colonized by aspen and willows. So ignore the silliness of this diagram, but just to get the point across, we have the smelter that has been huffing and puffing, and there's a lot of aerial contamination associated with it. So there's 5 big contaminants in the area: arsenic, lead, cadmium, and copper and zinc. And so these different contaminants, not only did they spread airily, but a lot of the contaminants ended up in the soil. And the more water-soluble ones ended up in the waterways and in the streams. And in Montana, there's a lot of, like, our field sites were on, like, in these wetlands. There's streams running through them. There's the Clark Fork River. So all of these metals ended up in the waterways as well. And it's really affected the ecosystem. So what we're doing is we're collecting blood samples from our 5 focal species, and we're looking at the heavy metal concentrations in our blood samples. And it's pretty interesting because some of their species feed on terrestrial insects, while some of our species feed on aquatic insects. And they'll pick up different contaminants based on where they're feeding from. And interestingly, even the species that feed on mainly terrestrial insects, they'll actually switch and they'll feed their nestlings aquatic insects and like grubs because those are really high in energy and nutrients that are really important for the development of their chicks. And so you actually see different metals in the blood from the chicks as you do from the adults. And what I really love about this project is that the data collected from this is actively being used to manage this land that we are like working on. Like it's actively being used to develop conservation strategies, and it's directly informing conservation, which was pretty neat. This is also what some of our sites looked like. On the right is that's actually one of our sites on the right. On the left, that's a portion of the Clark Fork River, and there were a lot of tree swallow boxes, which is a kind of separate project, but they had tree swallow boxes up around there. So this is just kind of some of the areas that we were working in. And this is a map of our field sites from the summer. So we have our 3 sites on the left, and those are all increasing distances away from the smelter. And as one would expect, the nests at Kagu, which is the site closer to the smelter, are picking up more of these metals, and there's more contamination and also decreased reproductive success of our songbirds. And then, so the reproductive success of the songbirds is also being used as an indicator of ecosystem health. And um, and so, yeah, so then we have a reference site that's kind of like way out to the side over there. And actually, there was contamination in that site, but it does look really pretty. Like it is being restored, and it's kind of the farthest one along that restoration journey. So, as we nest search, on the left is a screenshot of an app called Field Maps, and this is kind of what we use to collect all of our data. Each point on that map is a nest that someone found. The arrows are like clues, so you like cue into like, oh, did you see a bird fly with food? Maybe it's going to its nest to feed chicks. Like, did you see it carrying nesting material? Like, those are a clue to find a nest. But we have it color coded depending on the stage of the nest, so you can look at the map and you can tell, oh, is this nest? Does it have eggs in it? Are there chicks in it? How old are the chicks? Did we already bleed the chicks in that nest? Etc. Once we find a nest, we put flagging like 8 meters or a little more like meters away, and we take pictures so we remember where the nest is. And when they're too high up, we use a mirror to look into the nest and see what contents are in the nest. So nest searching was really fun. We'd sometimes see an adult on the nest. We sometimes see chicks in the nest. Chicks hatching. I caught this dusky flycatcher hatch day chick on the upper right. And then the bottom right, there's a warbling vireo chick, which we were banding. So in addition to our focal species, we were also kind of just monitoring these tree swallow nests as well. They put boxes up and down all the major rivers in Montana. So we were part of many different crews who were helping to monitor the tree swallows. So we would take pictures and weigh them and band them when it was time. We also got to join the banding team on there. We got to do on their field when they went into the field as well. So we got to help band some birds, which was very fun. But aside from field work, I also went on a lot of adventures. I hiked to this alpine lake, like 15 and a half miles. We saw elk outside of our house. I saw dusky grouse. My friend came and visited, and we went to Yellowstone together. We also saw moose at our field sites, which was really cool. We saw a bear. We saw Clark's nutcrackers, and I picked lots and lots of huckleberries. Huckleberries are a very big thing in Montana. And then my mom came at the end of the season, and we went to Glacier National Park together, which was absolutely beautiful. But above all, I met some really, really great friends during this project. This picture is actually from like an article that was written about the project, and so they mentioned kind of like what we were doing. But I made some really great friends during the summer, and I really fell in love with Montana. I'm just so grateful that I was able to be involved with that project. And Yeah, with that, I want to thank everybody. I want to thank everyone who's here, who came to listen to my program, who has supported me in the Audubon chapter for many, many years, and all these organizations and people who have really just helped me make all these projects possible and all of these experiences possible, which I'm so grateful for. And with that, I will take any questions.
Oh,
Thank you.
Violet, thank you so much for that wonderful presentation. I've been following this all along, and I actually feel smarter having listened to this whole thing just now. And as far as the bear in the title of your program, I know you and I saw a bear from a
Yeah,
distance in Montana, but I know you also had to carry bear spray every time you went out to do your work there. I know it wasn't easy living on Mandarte Island for
Oh,
10
I
weeks
loved it.
with no running water or
Oh,
electricity.
it was easy.
It was
I mean,
easy.
I loved it.
Yeah, you had to bring your own drinking water back with you. You had to take a couple of boats to go take a shower and do laundry. It was um you
That
did
was-
you did so well. Um let's see. We're gonna take any spoken
Yeah, feel free to
answers.
take yourself off mute and answer any questions, and then
Yes,
we move on
and then
to
I'll read through the
anything in the
chat
chat.
after anyone
There's
asks
anything
any
in the chat
questions.
for these questions. Yeah.
Hmm. Um why don't we start reading through the chat actually? Let's see.
Feel free to
JR,
interrupt and ask a question if you have one.
oh,
Oh, I,
sorry,
I'd
there's
like to
someone.
say something.
Sure. Didn't hear.
Yeah,
Oh,
this
there's the
is
chat.
great. I've loved science and nature my entire life. I'm so proud of you, and I've learned a lot, even though you were filling me in as you were going along. This organized material made things much clearer. I'm very, very happy to have enjoyed this.
Yeah, thank you. Thank you. I'm gonna answer Ted's question in the chat. Why can the city outcompete the song sparrow? Just the size. Yeah, the size and just they're more generally aggressive and they're the dominant species. Kate Johnson did her thesis on kind of how the fox sparrows moved in and started to outcompete the song sparrows. And she did feeding experiments during the winter time where she set up like arenas with seed and just seeing like displacement, like if the fox sparrow comes and the song sparrow like flies away, that's like a measure of that kind of dominance. And so they did 63 feeding experiments, and fox sparrows displaced song sparrows 91% of the time in those experiments. So, and also what I didn't mention is that, the fox sparrows are lining their nests with these feathers, and I think that they might be increasing their reproductive success because of that. Um, and that might be, and the song sparrows don't do that. So maybe that's another contributing factor to why the fox sparrows are doing so much better on the island than the song sparrows. Yeah.
I'll just read some of the comments from the chat and then we'll take any more questions. J.R. Murphy says it is incredible that you discovered that the white feathers created more antibacterial substances. Amazing
Yeah.
research, Violet.
What I would love, I didn't mention, but what I would love to do with that was like, I don't have the time to do this, but I would love if someone in the future like you could swab the eggshells of like the song sparrows and of the fox sparrows, and you could like measure like bacterial, like you look at the bacterial load on the shells, and you could also like swab nestlings and see like what bacteria is growing on the nestlings. I mean, if it is like that Bacillus like uniformis, I'm not sure. And or you could even like switch like feathers, like remove feathers from a nest and like do a cool experiment and like see how it affects the like microbiome of the nest. So that would be a cool way to move forward with that.
That would. Another
Thank you
comment.
for all the great comments saying thank you. And that,
Yes, maybe not everyone is seeing the chat, so I'm just going to
oh,
keep
Sandy has her
reading.
hand up.
Oh, sure,
Sandy.
Sandy.
Well, okay, can you hear me?
Yeah.
Okay. All right. So the feathers, the white feathers in the nest. So you've helped with the banding, and and anybody involved with me knows that the tree swallows line their nests with feathers.
Yeah.
And I never really know first the answer I give to that. Why do they do that? Is we think it maybe conceals the eggs, number one, or helps the eggs stay warm. never was it brought up that it had some thing that did the microorganisms or whatever you say.
Yeah.
We do know, I actually had eighth graders did a little test one time, and tree swallows prefer white feathers. We know
Yeah.
that when they have a chance to do F-fan. But this was amazing to have this whole other component of like the feathers do something, micro whatever.
Yeah, I know. I
Amazing.
saw that paper. Thanks. Like
Oh,
I saw that
well,
paper.
figure out why did the house sparrows have those feathers?
Yeah, I know. I know I'd seen like there was a study looking at like spotted starlings in Europe about it, but yeah, I'm not. I mean, there's a lot of variation in why I think the birds might use those feathers and the function that they do play in these nests.
Mhm.
But yeah, these feathers, I mean, I saw a tree swallow paper that was like, they prefer the white feathers. I think it might be because they have these antimicrobial substances and it would and the moss also, like green plant material, is also known to sometimes
Mhm.
release like antimicrobial substances, and I feel like the ones on Mandarte, those nests have a lot of moss as
Well, the
well.
chickadees have moss. The chickadees
Yeah,
use moss.
the chickadees in Montana had
Who
moss.
knew there was this other component? I just thought they liked the nice soft bed. I always say I'd like to be in a chickadee nest if I had to be in a nest, unless I could be in a Baltimore Oriole nest. But anyway, you're fantastic, amazing. I've known you since you're about 10,
Thanks.
and it's just fantastic
Thank you so much,
what
Dan.
you're accomplishing. And I
I
tried
really,
to follow you the best I could.
I really appreciate it. Um,
Say, Violet,
yeah.
this is Gail. I have a question.
Hi,
So,
Gail.
so I was fortunate, I grew up spending my summers up in Glacier National Park.
Nice.
And, so you're very aware of the smelters, obviously in Montana.
Mhm.
And, so I was wondering, you were talking about, you had that sort of a benchmark, site,
Mhm.
a little bit away from that smelter.
Yeah.
And do is do you think about, I guess this question has a couple, the predominant way the wind
Yeah.
flows,
Yes.
and is there a lot of testing that's done or a lot of studies that are done looking at the impact of, the natural wind direction, and also how over time, over time has that been reduced as the smelters have stopped, been shut down?
Yeah. So with the wind direction, that's actually one of the ways that Mary determined where her field sites would be located. And I'm unfortunately not as involved in that project as I am in like my own projects I'm working on. I kind of just like joined her like as a field technician, and we got a brief background on her project, but I'm not I'm not really involved in any of like the data analysis or super like conceptual stuff around it. But I know that she chose her field sites based on where the prominent wind direction was for the past 100 years. And I know that, the smelter has been, I'm not sure if they've looked at like it would be cool. Like they've looked at soil. They've also looked at like what metals are in the soil, and they did testing on the soil. It would definitely be cool to look at like soil samples and like the water samples from like when the smelter immediately like from like the
Yeah.
1980s versus now. Like that would be really interesting. But I'm not sure like if they might have that data. I don't know. But I do know that we are seeing still today all of these like impacts and what
Oh, I
they're doing.
bet. This
Yeah,
is, it's got to it'll last for
mhm.
decades and decades.
and
Yeah.
and so they're actually in some of these areas, I think they're literally removing soil. Like there's this big whole watershed committee, and they are really involved in the um conservation of that land. And they've been like restoring, they've been building like beaver dam analogs to try to get the beavers to come back, which is working. And I think they're actually like removing the soil and replacing it.
Yep.
Like now that you say that, it's jogging my memory, and I'm pretty sure that that is what they're doing. But yeah, I wish I was involved more in the project and could answer that fully.
Well, great. Thanks.
Yeah, yeah, of course. Um, Ted asked, has your work around the Anaconda region begin to result in changes in industrial practices in that region? I don't know the exact extent to which that may be the case, but I do know that the smelter stopped in 1984, and that there are a lot of conservation efforts and like efforts focused on managing the land and like restoring it currently. And like our, yeah, like our benchmark like reference site is one of those sites that is really like that has been this furthest along that path and like restoration. But I I would have to look into it more um to answer that. And are there only white feathers on the island? Yeah, so I mean, there might be like a cormorant feathers or something else, like some other bird feathers on the island, but there's not a whole ton of species on the island. There's also cormorants that nest on the island. There's also blackbirds that nest on the island, so I guess in theory they could use feathers from blackbirds if they wanted to, but the white feathers are on the island from the gull colony. And you know what's interesting is that the song sparrows don't use the feathers, and also of all the other fox sparrows I've looked at from these museums, they don't typically line their nests with feathers at all. So, I mean, it might be an antimicrobial thing, and it also might be them like opportunistically, when they have white feathers accessible to them, that they're using them when they can. Yeah, air sampling around the smelter should be archived and available over time. Yeah, I mean, I wonder if Mary's, I wonder how she's incorporating. I wonder how she's incorporating that. She might be incorporating it in some way or another, but I'm not really involved in the analysis of that project.
We had a comment there congratulating you on getting funding. I just want to
Yeah,
mention that that was
thank you.
not a given. There was some funding at school that was cut for many people. It's obviously a big issue these days, and it seems to need to be continually revisited, applied for, and it's a great thing that it came through.
Yeah, no, I really appreciate it, and I appreciate all the kind comments.
Yes,
Um,
Jean from Music
yeah.
Together from from 3 years old.
Yeah, I really appreciate the kind comments. Yeah, the funding, there's been like, yeah, there's definitely been a whole lot of funding issues, but yeah, Cornell, they sent me to AOS. They were able to give me funding multiple times for my projects. And so is, and through the lab and through cows. So grateful for that.
Thank you, everyone, for your very kind comments.
Oh, Bernie has his hand up.
Sure. Hi, Bernie.
Hello.
Bernie?
Can I ask a question?
Yeah.
Actually, two things. So the first question, and congratulations on all the stuff you've been doing, and it's super. So, first question is, what's the next phase in the island study with the fox sparrows and the song sparrows?
Yeah.
Whether you're continuing on or if someone else is going to be taking over,
Yeah.
what's the next phase of the research with those two species on that island?
Mhm.
And what do they yet need to know that they don't know yet? And then the second question would be, what will you be doing on your adventure down in Argentina in the
Yeah.
coming weeks?
Yeah, thanks, Rudy. Yeah, so all very exciting, but yeah, so so I work with John. His name was like briefly mentioned, but John is a postdoc that I've been working with since the spring on my genomics project. And so him and Catherine, who helped me with the nest paper, have been getting on board with Jen, and they've all been working really closely together, and they actually visited the island at the start of the summer season. And so John's very passionate about the fox sparrows, like now working on it with me especially. And so he actually wants to put trackers on them. He wants to put trackers on them, and so we'll actually be able to see like how if they're moving, because we know that they're resident individuals because they're banded, but we don't really know how exactly they're moving around the island. So that's definitely a direction. They're really looking at they're doing a lot of song sparrow stuff. They really love the song sparrows. So they're looking at the sparrows like around the islands and kind of like how like they're locally adapted to Mandarte and to those surrounding islands. But Yeah, I know the two undergraduates who are going out there next year. This is going to be continuing. So Peter RC's he did his PhD out there. He like built the cabins out there in the 70s and he ran the project for many years and now Jen's taking it over. So it's definitely going to be undergraduates heading out there following years and who knows what projects they'll be able to do. Hopefully they swab eggshells and do that. Um, but yeah, I'm so grateful. I got into a neotropical ecology course with Leo Campagna at the lab, who's also in the Fuller lab. And we're going to Patagonia over winter break. I leave on January 1st, first the Andes, and then Patagonia. We're flying into Buenos Aires and then going to Bustamante. So that's really going to be like us developing like our own research projects and we've been reading papers all semester about like projects that they've been doing out there and we've kind of been learning about the natural history of the area and kind of like what conservation issues are out there, kind of like looking at like conservation issues related to like the seabirds and the water and then because it's going to be really we're going to be on along the coast but then also like some like grassland species so it's really exciting because he's been taking us through like statistics and like how to like formulate questions based on like analyses so we're going to get to like develop our own projects and I'm not sure what my project will be yet. Maybe on a songbird. Like you could do it on anything though. It's not super. He's with the lab, but it's not super bird focused. So I knew someone who did a project on looking at insect abundance on different plots and like looking at species interactions. So that'll be a really cool opportunity as well.
And so they only chose 10 people for that course this year, correct? In past years, it was closer to it was 16 to 24 people. So that's been affected by the funding as well. So that should be an incredible adventure. I can't wait to see what happens. So any other questions, comments? By the way, I love all your beautiful photos, Violet. I know I'm your mom, but your photos and your slides are beautiful. I know I've seen them from time to time in different configurations. And all right, well, thank
Well,
you so
is
much,
anyone wait,
Violet.
we'll give them a minute if they have if anyone
Oh,
has
okay. We'll let them just decide if they
any
want.
questions. If not. Okay, sounds good.
Anyone.
Okay.
All
Okay,
right.
so hold it. So will you come back after your winter vacation and your research and tell us with equally great photographs what you saw or did?
I mean, I would love to do that. I would love to do that. I plan on taking lots and lots of photos while I'm there and go birding a bit. So I would definitely love to like share that project as well.
So, Mom and Executive Director, you've got the topic for a talk come March, April, May.
We will be catching up with Violet in the future, I'm sure. Keep your eye on her and we will have another catch up. Absolutely.
Super. Thank you. The best of luck to you, Violet.
Thank you so much.
Do we smell master's degree in the future?
Yeah, I'm gonna take a gap year. I want to go back to Montana, but I want to take a gap year and then I want to and then apply to grad school. So
All
maybe a
right.
PhD.
All right.
Good for you.
Thank you.
Thanks so much. This was just excellent.
Thank you.
Thank you,
Thank you,
Violet. We're
Violet.
so happy to have you present with us tonight. Really enjoyed it. Fantastic. And thank you everyone so much for being here and for your support. And we have a thank you, Violet, and to all a good
Oh,
night.
thank
Yay,
you,
Anthony. Thank
Anthony.
you.
Great job, Violet.
Thanks.
And I hope to see some of you on our upcoming walks and speaker programs. And wonderful to see you all. And take care and have a great rest of your evening. And the recording will be online pretty soon.
Good
All right.
night.
Good night.
Good
Good
night.
night.
Thank you, Violet. Thank
Thank
you,
you.
Leslie.